How you doing, guys? Today's lesson is going to be on DNA replication. So in the last lesson, we learned about the structures of DNA. We learned about some of the people that were important in the discovery of deoxyribonucleic acid. And we want to learn two things about DNA, two of its major functions. Obviously, the first function, like we said, was to be a kind of blueprint or a cookbook, like I called it in the last video, a collection of all of the recipes or codes for all the proteins that are made for you that give you your traits. Right? That's ultimately what it's made for. To make it really simple. DNA is nothing more than a collection of codes to make proteins. That's all it is. It doesn't actually do anything. Just like a cookbook doesn't do anything. A cookbook just sits there. It gets read by a chef or someone who's cooking the meal. DNA doesn't actually do anything. It's being read and being used by parts of the DNA. So two things that DNA is used for. Number one, one thing that DNA does is it replicates. And another thing that it does is it makes protein synthesis. Today, we're going to be focusing on the replication part. Okay, we're going to do protein synthesis in the next lesson. Today, we're going to be focusing on the replication part. So one thing that we haven't learned about yet was a process called mitosis. And we will be learning about that in the next chapter. I like to learn DNA first and what it is prior to learning about what mitosis is. That's just a preference of mine. Other teachers do it the other way around. Some people do mitosis first and then talk about DNA. I like to talk about DNA first and then do DNA. It's not really a big deal. I think you guys will get the drift of it. But mitosis, what mitosis is, let me go up to my whiteboard. What mitosis is cell division. Cell division. Okay, so one cell will actually create a clone of itself, and then split itself into two, and we call this the parent, and we call this the offspring. And these two offspring are genetically identical to the parent. Genetically equal to the parent. Okay. Now, the only way that's going to happen is if the DNA here, so if this was a human, it had 46 chromosomes, these 46 chromosomes have to be replicated into the cells of the offspring. Okay, so how that is done is essentially a cell will take its 46 chromosomes and it'll make a photocopy of them. Just like if you had, a textbook or a cookbook or whatever kind of book you prefer. And let's say you were absent for a particular assignment and you wanted to get the notes from a buddy of yours, you could borrow their notebook and take that notebook over to a Xerox machine and Xerox the pages that you missed for the day. And then you would have the notes that you missed, right? So if you took these 46 chromosomes and you made a copy of the 46 chromosomes, or you replicated those chromosomes, right? Because that's what you're doing. You're replicating what you already have. So you already have these 46, and then you replicate those 46 chromosomes, you'll have a total of 92 chromosomes, right? And 92 chromosomes we know is too much for the cell to have. The cell normally has 46 chromosomes. Right now it has 92. So what we do through this process of mitosis, which is called cell division, we are going to divide these 92 chromosomes that we just produced, that we just replicated from these 46, and we're going to take half of them, which is 46, half of 92 is 46, and we're going to put them into one offspring, and we're going to take the other 46 chromosomes and put them into this offspring. And what we've done was we've taken exact copies of these original 46 and placed them here, and we took the other copies, the originals, or a mixture of the originals and the copies, and we put them here. So if these were books, this book and this book are identical to each other, and they're also identical to that original book that you took from your friend, right? Because if you think about it, if you took this book from your friend, and then you went to the Xerox machine, and then you took all the papers back with you. In one hand, you would have the new copies that you made that came out of the machine, and you would have the original book from your friend. So you would have two copies of the notes, just like you have two copies of the cell. The only difference is that original book doesn't exist anymore. It's become part of the other two books. It's kind of like if you had a peanut butter sandwich and you cut that peanut butter sandwich in half. Okay, kind of in a way, maybe not, but you get the point. Okay, so what we want to learn today is we want to learn how do we replicate? How do we take these 46 chromosomes and make them into 92 chromosomes? Right? How do we take these? How do we take this notebook and make a copy of that notebook? Okay, that's what we want to talk about today. Right? So if we go back to our little lesson here. Okay, so before the cell divides, okay, before we go from that one original cell to those two new cells that are identical to the original, the DNA must be copied first. Okay, we have to take the 46. Let me clear that one sec. We have to take that 46 and duplicate it or replicate it into 92 before we split. Okay, we cannot split before we replicate, because if we split before we replicate, our new cells will only have half of the genetics that our original cell has, and they will not be equal to the original parent. And that's what we want. We want the offspring to be equal to the parent. Here, these are not equal to the parent, so that would not be a good thing. So before the cell divides, DNA must be copied first. Okay, so that's really important. So what we learned in the last lesson was that there are double helix, right? There's two helices, and one side of the helix has nucleotides, and the other side of the other helix on the other side has nucleotides. And these nucleotides have phosphates and ribose sugars, and they have nitrogen bases. Okay. We also know that these bases like to bond to other certain bases, A to T, G to C, T to A, C to G, right? And we call these base pairs, if you remember from the last lesson. We call these complementary strands. So this, let me just change the color here. This strand is complementary to this strand. So strand 1 is complementary to strand 2. Okay, A is the complement to T, G is the complement to C, and so on and so forth. So we call these complementary strands, right? This A to that T is complement, and so on and so forth. So AGTC is the complement to TCAG, and vice versa. Okay, so in order to duplicate or replicate, right? In order to replicate this DNA, we have to know what the letters are. We have to see the letters inside of the DNA so that we can replicate what the letters say, right? Like if you wanted to copy your friend's notebook, you'd have to open the book to the actual page that you want to replicate so that the Xerox machine, the copy machine, can scan the actual letters that you want copied, and it will spit out a replicated page of those letters, correct? So that's what we want to do here. We have to open the book. So the thing that we want to talk about now is we want to talk about enzymes, right? Enzymes are proteins. Enzymes are proteins that catalyze reactions. Okay, they help reactions go. Okay, so we're going to need enzymes here. And some of the enzymes that we're going to need, one of them is called helicase. Helicase. Okay. And another one is called polymerase. Helicase is going to unwind our helix, or unzip. Unwind / unzip the helix. We can't read the information when it's all squiggly. Okay, we can't read it when it's all squiggly like that. We have to unzip it. We have to unwind our helix. Okay, so we want to go from something that's all squiggly like that to something that's open like that, kind of like a zipper. That's why we call it unzipping. So if you think of a zipper on a jacket or a coat or something like that, if you've ever zipped up your coat and then the zipper breaks and the bottom kind of opens up and you can pull it apart on each side, that's kind of what we want to look at, right? Because we want to unzip this. Now I have a picture up here. Let's see if I can see which one. Here it is. Okay, so here's my double helix. We can see my double helix over here. And then here is the zipper part. This is the part that's being opened. Okay, don't worry about what's going on here yet. We'll talk about that in a minute. I just want you to see how one side of the zipper is opening up here and the other side of the zipper is opening up here. Okay, so we want to unzip that. And that's what helicase is going to do. Helicase is going to unwind or unzip our DNA double helix. Okay. Now, one thing that we have to know about DNA is the structure and what we call the reading frame. Okay. We'll get back to polymerase in a minute. Okay. So let's talk about something called the reading frame of DNA. DNA is said to be read in what we call a 5' to 3' direction. Okay, so when you open a book and you read a sentence, you read that sentence from left to right. That is your reading frame, left to right. You're reading these words, reading frame 5' to 3' from left to right. You are not reading this from right to left. You are not saying 3' to 5' frame reading. You're saying reading frame 5' to 3'. That's what you're reading. So you're left to right. Okay. DNA can actually only be read because what it is, it's instructions, right? DNA is an instruction manual, and it can only be read in what we call the 5' to 3' direction. Now, what reads DNA? What is the thing that's actually reading DNA? That's the polymerase. Okay. Polymerase is going to read, or your DNA, it's going to read the letters A's, T's, G's, C's. Okay. And it's going to read these letters and it's going to produce the complementary strand. Okay. So polymerase is going to see an A, it's going to read an A, it's going to kind of manifest a T. I don't want to get too much into it. We're going to get into that later. Okay. G is, it reads a G, it's going to manifest a C. It reads a C, it's going to manifest a G. It reads a T, it manifests an A, so that it makes a complementary strand to what it's reading. Okay. So now what does 5' to 3' actually mean? Okay, let's take a look at this picture that I have up here. Okay, this picture here is showing you a ribose, or it's showing you a nucleotide, right? This is a nucleotide. So this base could be A, it could be T, it could be G, it could be C. It doesn't matter. I'm just going to pick A for argument's sake. Here is my ribose. Okay, and here is my phosphate. So we have a nucleotide. That is exactly what we looked at in the previous lesson. Okay, so that's my nucleotide. What you see here is numbers 1, 2, 3, 4, 5. Okay, the number 1 stands for the first carbon in this particular molecule, right? So chemists, when they name compounds, a lot of times they have to use numbers to tell the person who's reading what molecule or what atom they are talking about in a molecule. So, for instance, if you looked up the back of a ingredient label on an item of food, or especially like a shampoo bottle or maybe a toothpaste container, it will have all the active ingredients in the particular bottle. You know what? I'm going to pause for a second. I'm going to see if I can find an example of this. Hold on one sec. Okay, so I found an example here. Okay, so if we take a look right over here, we see this really long word. Okay, but we see, I don't even care that what this word says. It says cyclohexanecarboxylahide or whatever it says, right? Diide, whatever. It doesn't matter. What I want you to see is this 3. Okay, that 3 is talking about a specific molecule in the, or specific atom in this giant molecule. Okay, that number 3 is important for this naming. Okay, so the same thing is going to go for this particular, where's my picture? For this particular compound, this nucleotide. So this number 1 stands for carbon number 1. That is the first carbon in this molecule. And it's always going to be this carbon. If we flip this thing upside down, okay, that carbon will follow. And so if I just took this and rotated it 180 degrees, this carbon would end up on this side of the molecule. So if I mirror image this, if I do something like this, okay, this will be the number 1 carbon, right? This will follow and be on this side. This carbon, no matter what orientation it's in, will always be in that position, if that makes any sense to you. Okay, so this is the number 1 carbon. This is the number 2 carbon. This is the number 3 carbon. Here's the number 4 carbon, and here's the 5 carbon, right? So we call them prime. So this is the 1 prime, 2 prime, 3 prime, 4 prime, 5 prime carbon. So now if we look at this picture, we can see that on one side of the DNA, our little pentagon ribose sugars are pointing in one direction, okay, where the tip of the pentagon is pointing up on this side, and the tip of the pentagon is facing down on this side. And the reason that is because Watson and Crick, one of the things that they discovered in their discovery of the shape of DNA is that they discovered that DNA runs in what we call anti-parallel forms. Okay, what that means is that one of these strands, one of the helices, runs in one direction, and the other helice or the other helix has to run in the opposite directions. They're anti-parallel. Okay, they go in opposite directions. Okay, so this one is the 3 prime. What that 3 stands for here is the 3rd carbon, right? This is carbon number 1, carbon number 2, carbon number 3. Carbon number 3 is at the bottom, right? Carbon number 5 is at the top. And over here, carbon number 3 is at the bottom, carbon number 5 is at the top. Over here, carbon number 3 is at the bottom, carbon number 5 is at the top. On this side of the DNA, on the opposite side of the DNA, the other complementary strand, the 3 is at the top and the 5 is at the bottom. The 3 is at the top, the 5 is at the bottom. The 3 is at the top, the 5 is at the bottom. Because what we've done is we've taken this and turned it upside down and put it on this side. But all the carbon, the names stay in the same spot, right? So this carbon number 3, which would normally be here, if we turned it upside down, it goes to the top. So we call this anti-parallel. Now, the reason I bring this up is because polymerase can only read DNA in 5 to 3 prime directions. Okay, just like you can only read left to right on a page, or if I wrote words, you could only read those words and have them make sense to you in the left to right direction. Polymerase can only read in the 5 to 3 prime direction. It cannot read 3 to 5 prime. Okay, So that's going to become very important, because the first thing that we want to do when we want to replicate DNA is we have to take the DNA apart. Okay. And when we take the DNA apart, okay, it's going to look very much like this. Okay. We're going to have, here's our double-stranded, our double helix, and it's going to open. Whoops. It's going to open up. Okay. And one side of the zipper is going to go this way, and the other side of the zipper is going to go this way. And here's the thing. One of these is going to be the 5' end, and the other side is going to be the 3' end. Okay. In this particular case, this is the 5' side, and this is the 3' side, because that's just the way it has to be, right? Because one side of this has to be running in this direction. The other side has to be running in the opposite direction, because they're anti-parallel. Okay. Why are they anti-parallel? Okay. Let me get into that as well. The reason they're anti-parallel is because what Watson and Crick did, whoops, I don't want to do that. Hi. Okay. What Watson and Crick did was they found out that if it's impossible for DNA to run both sides in the same direction. It's absolutely impossible for both of them to run in the same direction, because the C would never meet up with the G, and the A's would never meet up with the A's in the right form. They would be off a little bit. If I flip this upside down, okay, or if I flip this, or right side up, whatever you want to say, if I made them both go in the same direction, the G would be like slightly lower than the C, and the A would be slightly lower or higher than the T, and they would never meet up properly. See how here they're nice and level with one another? Okay. If I flip them, if I made them both go in the same direction, this G might be higher or shorter than the C. The C might be up here instead of over here, and they're not going to bond properly. Okay. And if I did that, this A might be too high and it might not bond with the T. Well, Watson and Crick, one of the things they found out was that you had to flip one side in order for the C's and the G's and the A's and the T's to line up perfectly together, right? So they did a lot. They found out, one, that they're anti-parallel. They found out, number two, that they read 5 to 3. And they found out, number 3, that they have to twist, right? We learned about that in the last video. Okay, they have to twist in order to go up to form this double helix. Okay, so when we do essentially open the DNA, okay, once we unzip it, because that's what helicase is doing. Here's that helicase, right? That helicase is unzipping my DNA. It's starting from the end, because your DNA is linear. Your DNA has a beginning and it has an end, right? This chromosome has a beginning and it has an end. Okay, we're just going to say that this X represents the beginning, and over here, represents the end, end. Okay, and what's going to happen is this helicase has to open the zipper at the beginning, and when it does, it's going to expose the two strands. It's going to expose the 5' strand, and it's going to expose the 3' strand. Okay, now when it does, when these two strands are exposed, we call this a replication fork. Okay, and you can kind of see why that's called the replication fork. It kind of looks like a fork with one less tongue hanging out of it, or whatever it's called, right? So this is the replication fork. Okay, so the helicase here is going to open up the zipper, or unzip the zipper, undo the helix, and then our polymerase is going to come in and it's going to read the letters in the 5' to 3' direction. So this is the 5' end, and that goes all the way up to the 3' end over here. And this is the 3' strand, and that's going to go all the way up to the 5' end over here. Okay. Now here's the thing that's very special about replication. The polymerase, like I said, can only read 5 to 3. So it has to go in this direction, 5' to 3' direction. That's what this arrow is about. Okay. It's able to do that just like you are able to open a book and read, right? So the polymerase will start here. If it sees an A, it makes a T, right? If it sees an A, it makes a T. If it sees a G, it makes a C. If it sees a C, it makes a G. If it sees a T, it makes an A, and so on and so on and so on. Every time it sees a letter, okay, A, T, G, C, it makes the complementary letter to that, right? So if this was an A, let me get a different color so you can see it a little better. Okay, if this was an A, it would make a T. If this was a T, it would make an A. If this was a G, it would make a C, and so on and so forth. We call this the leading strand because polymerase can start right at the front and work its way all the way continuously until the end. Okay, as this zipper opens, this polymerase is just going to continue reading letters and making new ones. Okay, so we call that the leading strand. Now, the other strand is what we call the lagging strand. And it's the lagging strand because it's not able to be read 5 to 3. This was 5. This is 3, right? The 5s are over here somewhere, right? The 5 is over here. So what we can do, it's almost like we're opening the book from the back, right? Over here, we open the book from the front, right? We open the book from the front because that's where the 5' is the front of the book. The 3' is the end of the book. So it's almost like we're opening the back of the book, right? If you took a book and you opened it from the back cover, right? What you would have to do is you would have to read, and you're only allowed to read from the front of the chapter to the end, right? You can't read from the end of the chapter to the front. You have to read from the beginning of a chapter, and you have to read to the end of the chapter. Here, we can start on page 1 and work our way to page 100, right? But here, we have to start at 100, right? We open the book and it's on page 100, but let's say that chapter begins at page 80, right? So we can't start at 100 and work our way to 80. We actually have to wait until the book is opened to 80, and then we can read from page 80, and we can then read to 100. Does that make sense to you guys? Okay. We have to wait till that chapter is all the way opened before we can start the chapter and then read to the end of the book. So you're reading the end first. And then as you're reading chapter, page 80 all the way up to 100, during that time that you're reading, more pages are being opened up. So up to page 60. So by the time you're done with 100, by the time you're done with chapter, the last chapter, the second to last chapter opened. So when you're done with this chapter, you can then go over here and start reading up until page 80 where you already read. And then you can stop once you get to page 80. And while you've read page 60 to page 80, page 40 to 60 opened up. So once you're done, once you get to 80, then you can go to 40, and then you can read from 40 to 60. And then once you've read, while you're reading 40 to 60, pages 30 to 40 opened up. And once you're done, once you stop reading here because you read all that already, you don't have to read it again, you've read it all, okay? You can go back to page 30. So you're reading the book one chapter at a time backwards, if that makes sense to you. And that's what your DNA has to do, okay? Once a little piece opens up, so here's the 3' end. Once a tiny piece opens up, you can start at this 5' carbon, which is on this side, and you can read until the end. And that's what this is. We call it the lagging strand because this side has to occur in pieces, right? It occurs in pieces. It does a little bit, and then while that's happening, more is opening up. So while this polymerase reads these letters, even more DNA is opening up because of the helicase. And once it falls off over here, it can then go back to the open section, that newly open section, and read more. And as it's doing that, even more is opening up. And then it can go and do that. Now, you would think, and even by the name lagging, it almost seems like it's happening slower, right? If you started these two polymerases off at the same time, you would assume that the leading strand is going to finish. You would assume that this leading strand is going to get from point A to point B quicker than this one is going to get from point A to point B. But it actually doesn't, okay? The lagging strand and the leading strand are pretty much on the same pace. It's just that this strand, the lagging strand, has to occur in portions Okay, so it does this portion, then it goes back. Let me get a different color. I had to choose that color. Okay, it does this portion, and then while it's reading that, another portion opens up and it goes back, and then another portion opens up and goes back, and another portion opens up and it goes back. Okay, where this one's just chugging along, all the way down to the end. Okay, and then when it gets to the end, it falls off. Once that happens, what you end up with is something like this, right? So if I have a piece of DNA, and I have another piece of DNA like that, okay? And these are, what's going on? Excuse me. Let's get rid of that. Okay. And these are connected to one another. And then we start forking, right? We start getting our DNA replication fork over here, right? What's going to happen is we're going to have our polymerase reading from the 5' side to the 3' side, like this, okay, in this direction. And that's going to make the complement side to this. And then we're going to have our other one working on this direction, making the complements over here on this side. Eventually, both of them will eventually get to the end, right? And when they eventually get to the end, what I'm going to have on this side is I'm going to have by the time they fall off, what you're going to see is on this top side, you're going to see a new piece of DNA with a portion of the old original strand. I don't want to call it old, but the original strand, the red strand. And the complement to that red strand is going to be brand new. And that polymerase just made that complement. And on the bottom, we're going to see the same thing where half of that DNA is original, the red portion, and the other portion, the green portion, is the complement that was made by the polymerase going in the opposite direction. Eventually, by the time this gets all the way to the end, and the polymerase on top falls off and the polymerase on the bottom falls off, right? This blue polymerase will fall off, and this green polymerase will fall off over here eventually. Actually, it'll fall off over here because it's going in that direction, right? It'll fall off on this side, okay? But eventually it'll get all the way to the end, and what you're going to be left with is 2 copies, right? You've replicated DNA. Here's one replicated piece. Here's another replicated piece. And each one of these is equal to each other, okay? This information is identical to this information, right? Because you've copied it. And this, and these 2 strands here are identical to what the original used to be. But this original has now become part of the new one. That's what I said at the beginning. Like, your friend's book does not become part of your book, right? You have, new fresh pages after you photocopied it. So you still have your friend's original book, and then you have your photocopies. That's really the only breakdown of my analogy, is that here the original book becomes half of the new book, or half of both new books. Okay. So why don't you watch this video, and I'm gonna kind of commentate while this video is being played. Let's see if I can do that. See if I'm allowed to draw over this as it's going. Oh, I can. How nice. Okay. I'm actually going to reduce the volume so that I can talk while it's going on, and hopefully it'll work out. So here we have. Let's see. Here we have our DNA, double helix, twisted, okay? We need this book, and I'm going to be pausing intermittently. So we need this information. All of this stuff is the information, right? These are all A's, T's, G's, and C's. One side is complementary to the other, right? This side is complementary to this side. And one of them is going to run in 5' to 3'. One of them is going to run in the 3' to 5'. We are not sure which one it's going to be yet because of the video. Okay. But what we need to do is we need to open this book. And that's what's happening, right? That helix, if we go back a couple of seconds, was really twisted. It was a double helix. We need this book to be opened. So we're going to unwind it using helicase. Okay. So here are my complementary base pairs. And we want to replicate it, right? We want 2 pieces of DNA that are exactly identical to this one piece. We want to start with this one piece and we want to make 2 identical pieces. Okay, so here's my 5' end, here's my 3' end, right? So that means that here's my 5, here's my 3. Polymerase can only read from this point to this point. So that means this side must be the 5' end of that and the 3' end over here, right? So this top strand is going 5 to 3 left to right, and this bottom strand is going 5 to 3 right to left, anti-parallel, right? Anti-parallel. Okay, here's my helicase. Okay, my helicase is moving along the DNA, unzipping my strands and this is our replication fork. This is creating our replication fork. As this gets unwound or unzipped, we need to start copying it, right? So this was the top strand, okay? Don't worry about primase. Primase this is a primer. This is going to tell the DNA where to start copying. Okay, we don't really need to know that great a detail right now. But a primer tells the polymerase where to start copying from. Okay, because sometimes we don't want to copy the entire thing. Sometimes we only want to copy portions of it. So here's my polymerase. Here's my enzyme that's going to actually make my copies for me. Okay. Now polymerase doesn't actually make the A's, T's, G's, and C's, okay, which is important to know. The A's, T's, G's, and C's are not made by the polymerase. The A's, T's, G's, and C's come from your diet, okay? We get all these A's, T's, G's, and C's from eating other things that have DNA, right? So if you eat plants are made of cells. If you eat meat is animals that are made of cells, cows, fish, pigs, chickens, all those types of things. If you eat something or a fungus, like a mushroom, right? If you eat anything that was once living, that once living thing has cells. Those cells have nuclei. Those nuclei have DNA. Okay, so when you eat a cow, okay, and you eat a burger, okay, the cow's DNA is being ingested by you, and you are breaking down that cow DNA into A's, T's, G's, and C's. It's almost like if I gave you a Lego house already built, and you took the Lego house apart into its individual blocks, right? And you separated those individual Lego pieces, and you put each individual Lego piece into its own little bucket so that you could use those Lego pieces later. The cow's DNA does not become part of your DNA. You do not have cow DNA, but you're using the components of cow DNA to make human DNA. Just like you can take the Lego house, take it apart, and then build a Lego ship. Okay, that ship is not a house, but you use the parts of the Lego house to build the ship. That is exactly what's going to go on there. Okay, so the polymerase reads, right? That's what the polymerase is doing. The polymerase is reading the letters, and then it's calling, or it's going into the bins to get the complementary strands. Okay, and there you go. Here's the 5' end of the complementary side, and it's going to read every letter, and it's going to make the complement. And it's going to be the leading strand because it's going in one fluid motion. As this opens up, okay, so let me pause it. As this helicase opens up my DNA, this leading strand is just going to keep going along, chugging along. Okay. The other side, which is called the lagging strand, let me go back a couple of seconds. Okay. This lagging strand has to be done in parts. Okay. So this portion, here's the primer. That's where it was told to start. Don't worry about Okazaki. We'll talk about that. That's AP Bio stuff. Okay. We're going to read. Okay. Let me pause it real quick. This is the 5' end of the complementary strand, and the 3' end of the complementary strand is over there. So it can read from 5 to 3. So it read this portion already, and as it was reading this portion, the helicase was unzipping this area. And by the time it was done, this polymerase, or what we're going to see, the polymerase in a minute, is going to come downstream. That's what we call this, downstream. And it's going to tell the polymerase to start copying here to fill in the gap. Okay. So let me show you that. Okay, there's our new primer. And our polymerase is going to come. And it's going to fill in this gap. And while this is happening, more DNA is opening up downstream. And the primer is going to come back. And that primer is going to tell the polymerase where to attach to. And then it's going to fill in the gap. Now, this whole time, the leading strand is going in the other direction. Okay? All right, we don't want to know about exonuclease. Don't worry about that. Okay, we'll stop the video there. Okay? But that's the gist of it. So one strand, we call the leading strand. It's going to be copied continuously from one end to the other because it's reading the 5' to 3' direction. The other side is called the lagging strand because it has to be done in segments. Okay? There's a video here. It is of, you know this guy. I'll show it to you. I'm not going to play the whole thing for you. If you want to check it out, you can. Okay, it's what's his name? Crash Course, I think is the name of this video. Okay, if it ever loads up. Okay, and he's pretty good. If you just type in Crash Course DNA Replication, it should show you that video. There's an ad. There you go. DNA Structure and Replication Crash Course Biology number 10. If you want to look it up, that's the video you can look up. That's the video that's linked in the Versal lesson. Okay, and as always, there is a Quizlet. Okay, feel free to work on that. Again, feel free to change the mode to whatever suits your needs. Okay, flashcards, test, matching column. Okay, you can do whatever fits your needs. You can make it a game. You can make it a test if you want to make it look like more like a test. Okay, and don't forget you have a quiz coming up that will be posted. Make sure that you check the times for that. It has to be taken by 11 p.m. Okay, so thanks for hanging out with me today, and I will see you next class for protein synthesis. See you later, everybody.